Hello, and thank you for watching my talk on whether it takes a community to save a species, investigating community interactions among at-risk unionid species in the Great Lakes Basin. My name is Roland Eveleens, and this is part of some ongoing mussel research that I'm doing at the University of Windsor, located in the traditional territory of the Ojibwe and the Free Fires Confederacy of First Nations. For my master's thesis, I'm looking at freshwater mussels because they're a diverse species group with many community interactions and in need of conservation action. Within the lower Great Lakes, there are 41 native species of unionids, and 16 of these mussel species are federally listed as at risk of extinction from stresses such as habitat change, invasive species, and pollution, and freshwater mussels are disproportionately threatened globally. So I'm investigating what community interactions need to be intact to allow successful mussel conservation and restoration. Particularly here, what the contributions of trophic and community interactions to freshwater mussel populations and communities are to inform restoration and the potential for using more ecosystem-based approaches. For the conservation of mussels, we first need an understanding of which species occur together and where in order to direct restoration actions. This is especially important for mussels due to the many community interactions they're involved in and the overlap of species habitats leading to many species being found together at the reach scale. So firstly, we need to understand environmental and biotic features that shape, filters that shape local community assemblies. For example, the occurrence of suitable habitat conditions or host fish leading to mussels being found at a particular site. And then we also need to understand the trophic interactions that occur between mussels and other species groups, such as benthic invertebrates and fish. Now, degraded communities are likely to be missing species and so lack the interactions needed to support mussel populations. So restoration seeks to restore these connections to recreate self-supporting ecosystems. To do that successfully, we need to understand these interactions. Host fish to mussel relationships are already well studied, so I'm particularly interested in the interspecies interactions between mussels and the interactions between mussels and benthic invertebrates. For that, I'm looking at the associations between mussel species, patterns of mussel community assembly over environmental and size gradients, and the link to benthic macroinvertebrate communities, specifically investigating differences between subwatersheds, differences between tributaries and mainstem sites, and the relationship of mussel communities, mussel species at risk, and benthic macroinvertebrates. And so I'm presenting some preliminary results of this today, and I'm going to refer back to these icons for each of my aims as I work through my results. This work is based in the Sydenham River in the North American Great Lakes. So this flows into Lake St. Clair and is a hotspot of mussel diversity within Canada, featuring species at risk such as kidney shell, raid bean, snuff box, round pigtoe, northern riffle shell, and more. And so I was lucky enough to still be able to do fieldwork in 2020, although our data set is slightly limited. Now, we were able to sample 15 sites spanning 3 subwatersheds of the Sydenham River, ranging from 3rd to 6th order. Most sites were sampled for mussels and benthic invertebrates, so those are green dots on our map, while some used existing recent mussel data collected by other organisations, shown in yellow. We were also fortunate enough to be able to work with the Canadian Department of Fisheries and Oceans and the local St. Clair Region Conservation Authority to obtain other existing mussel data for the region. Now, for our own fieldwork, we followed a hybrid survey protocol involving a 4.5 person-hour time search combined with 10 1 metre square quadrates if we found an abundance of mussels or species at risk at a site. And then we also sampled invertebrates and environmental data following Ontario provincial methods to produce data sets and analyses that were more widely applicable. To just touch on the effectiveness of our hybrid protocol, for the 10 sites that met our criteria to be both surveyed by time searches and quadrates, we found time searches were more effective at detecting species than our randomized quadrates. But adding quadrates was beneficial because larger and more diverse sites, the time search didn't detect all the species that we found through the combination of both methods. And so to give context on the differences spanned by the sites we surveyed, these covered from large and slow moving and silty in the North Branch to smaller and still quite silty on Black Creek, while the East Branch was more flying with coarse substrate and then smaller and slightly more silty when we got into the tributaries, which is shown by the letters on the inset map here. Now, as I'm lacking the environmental data to robustly define a continuous gradient of environmental impact, I'm grouping my sites into categories based on subwatershed and watershed position to compare river main stems with tributaries, where tributaries are defined as having a stream order of 4 or less. And for these categories, we observe differences in waterway size, water chemistry, especially DO and pH, shading and channel characteristics, as well as sometimes differences in sediment compactation. So we investigated this using a PCA to sort of demonstrate these groupings. So here we're showing the differences in environmental conditions between sub-watershed. And while this PCA only explained a moderate amount of variation between our groupings and like riparian land cover, it still supports our approach of grouping sub-watersheds into different categories. And that sort of remains when we look at the difference between main stem and tributary sites. And so it sort of helps support our groupings of as waterways position and sub-watersheds. And then when we looked at the mussel species richness, we found clear patterns in a greater spread of species richness in main stem sites, and the East Branch sites were more diverse than the North Branch and Black Creek sub-watersheds. And that pattern remained when we combined North Branch and Black Creek to equalize sample sizes. And here I'm using asterisks to denote significant differences between catchments when tested using an analysis of variance. Now these patterns remain present when we just looked at species likely to be of interest to restoration, that being classified species at risk and species listed as being of special concern, and that these were only present in main stem sites and more diverse in the East Branch. And that pattern remained for species at risk alone, although sample sizes became too small to actually test the statistical significance of these differences. So it was because species at risk were only found at a few sites. And so therefore this supports significant difference, that we have significant differences in the diversity of main stem sites compared to tributaries and between sub-watersheds. And then when we look at the differences in overall community composition, we see these patterns remaining. So here, looking at the differences between the community composition of sites across sub-watersheds, this ordination combines the abundances of each species found at each sites, so that the closer points are together, the more similar the community composition of those sites are, with colours of points showing the different sub-watersheds. And we found that tested using a Permanova, these groupings were statistically significantly different. And that similar patterns remained when we looked at main stems compared to tributaries. So both supporting my first hypotheses of sub-watershed differences and differences between main stem sites and tributaries. Now these differences were mainly driven by the diverse assembly of species we found at large East Branch sites, such as snuff box, purple woollyback, northern riffle shell, and red bean, while our second axis was strongly driven by the more sediment-preferring species, with differences in species such as the occurrence of Wabash pigtoe or fat mucket and maple leaf. But then when we looked at where species at risk in particular were present, we found they occurred at specific sites. And so this is now serving as a springboard into my ongoing analysis beyond the preliminary results that I'm sharing here, to look into the environmental and species patterning driving the occurrence of these species at risk, as well as if there's any relationship between patterns of benthic invertebrate data and this occurrence of species at risk. And then finally, to link back to the conservation of species at risk, I also examined the relationships between species richness overall and the occurrence of species at risk, and where I found that species richness overall was much higher at sites where species at risk were present, and that the richness of all species explained much of the variation in the richness of species at risk found at each site. So therefore, there's saw that we had distinctly different species assemblies across sites, and while we already know that environmental conditions and host fish presence play a role here, the occurrence of species at risk at only some sites with diverse mussel communities suggests that biotic interactions either are or were present to support that occurrence of species at risk. And so I'm continuing to unpack any patterns between species, as well as between mussels and benthic invertebrates. And the conservation of implications of this will help address mismatches and sort of connecting spatial processes and gaps between research and practice that were evident from a separate systematic review of mussel restorations that I'll hopefully be publishing as a paper soon. But some points that were new to the Sydenham are the effect of sub-watershed and watershed position on mussel community composition, which can be used to direct the location of conservation and restoration actions, as well as the occurrence of species at risk in diverse assemblies, which suggests that maybe habitat quality is more important than specific conditions to support the co-occurrence of multiple species, as well as maybe the potential to need to translocate multiple species together rather than just a single species. And then further, there is potential to harness biomonitoring data more widely for this sort of community analyses. And so I'm looking forward to attempting to address these points further as I continue my analyses and seek to support the restoration of freshwater mussel species at risk. So with that, thank you for listening. And I'd like to thank my other committee members of Dalen Warno and Trevor Pitcher, as well as the supportive organisations, including the Department of Fisheries and Oceans and the St Clair Region Conservation Authority, as well as funding from NSERC and the Fishcast program, as well as all these lovely people that continue to help me with my project and the landowners that allowed access for our fieldwork. I'm happy to answer any questions, so feel free to get in touch through Twitter or email.